Good morning, everybody, and thank you, Jerry, for the invitation. And it's an honor to be here. I almost went to this conference when it was in America. Never did. And here I have to come all the way to Bulgaria to talk to you guys, which is great. I am going to talk about my research with water, obviously. I have my company, Quantum Biology Research, which is the basic research division, and I recently have an innovative biophysical technologies division, which is for contract research. So my title uses the word subtle energy, and I thought I should define it because maybe people are not familiar with this kind of electromagnetic energy. Unlike the electromagnetic energy that Moray King was talking about, this kind of energy is non-classical electromagnetic fields, is the term that I use because there are many terms out there. The scalar is a term that many people are familiar with, but that's only one of many terms. And now the million-dollar question is, are these all the same energy, or are they different types of subtle energy? Here's some characteristics that distinguish classical and non-classical. The primary distinction, do I have a pointer? Do I have a pointer? Just in case. Oh, yes, sir. Is there a printer? Yeah. The primary distinction being that classical electromagnetic fields are force fields and they exert a physical force on an object, whereas non-classical fields do not involve force. They are non-linear, they converge instead of diverge. They go backwards in time instead of forwards in time. They contain phase information and rather than frequency information, and here we have the differences. Okay, these can be generated by certain special kinds of coil windings, caduceus coil, a Mobius coil, and a BIFILLAR coil, and a toroid. Cyril Smith and Mei-Wan Ho use toroids, which trap the classical electromagnetic field inside, and on the outside you get a non-classical field. In contrast, these three kind of coil systems buck the electromagnetic fields by forcing them 180 degrees, and the actual force creates interference patterns which generate a non-classical electromagnetic field. So, this slide everybody's aware of, that classical electromagnetic fields have biological effects which are mediated by water. Obviously, you guys know all that. But it turns out that non-classical electromagnetic fields also cause biological effects, which, according to my research, are also mediated by water. Which means you can treat the water with these fields and then give the water to the biological system. So, biological effects of non-classical electromagnetic fields, historically speaking, actually was first discovered by Pat Flanagan, and then Andrei Puharich, and then Eldon Bird, and then myself, and Mei-Wan Ho, and others have subsequently, more recently, verified that these non-classical fields do have biological effects. So now the question is, are these effects also mediated by water? So my first experiment to demonstrate this was done by a colleague who works for a company called Dynamic Engineering. We used a very complex input. The electromagnetic signal is very complex. It's one wave riding on another wave, and there are many different frequencies involved. And this signal is then fed into a caduceus coil, and it was used to treat water directly, and the water was then given to human lymphocytes grown in tissue culture. The growth was measured using standard biochemical techniques, tritiated thymidine incorporation into DNA. And you can see that the direct effect caused a very large stimulation in the growth when the cells were treated directly, and when the water was treated and then given to the cells in culture, there was a significant effect, not quite as large as the direct effect, which I guess you kind of expect. And the concept, the explanation or interpretation of this is that some of the information is lost. The water does not store all the information. Okay, so now, as far as my original research, I have, being a biologist, I tend not to invent my own devices. Everybody, many people here have such a device for charging water, structuring water. So why reinvent the wheel? So I have used all of these various, right, white on white doesn't work so well. I've used various commercial devices which generate both classical and non-classical electromagnetic fields. Yeah, I did. Yeah, but I don't see it because I'm colorblind, so I don't see it. Another one. Yeah. This one brighter? Oh, yes, this one's good. Okay, right. So my primary interest, of course, is non-classical fields, and I've worked with technology from these different companies. And then recently I'm working with my own simple signal generator and a amplifier and a caduceus coil or a BIFILLAR coil, which we'll hear more about as we proceed. So in my experiments, I work with different sources of energy, classical and non-classical electromagnetic fields, geometric patterns, two-dimensional and three-dimensional patterns, also generate non-classical fields, acoustic energy, gemstones, bioenergy, and quantum fields generated according to Siki from a Mobius coil. I'm going to focus on the non-classical electromagnetic fields. I have done a lot of work with healers, but that's beyond the scope of this lecture. As far as biological targets, well, targets in general, I either work with living cells grown in tissue culture, the DNA molecule, or the water molecule. But I'm going to focus on the water because I don't have time to discuss all the other research. Now, as far as measurements, I use many different techniques to measure the changes in the properties of water. I'm going to talk about most of these today, and probably won't have time for the simple pH measurements. So let's start with measuring the optical properties of water. I'm doing this historically in the sequence that my original experiments were done with ultraviolet spectroscopy because I happen to have one, access to one, and the point being that ultraviolet spectroscopy measures the excitation of electrons in trapped gas in the water, oxygen primarily. Now, the region of the ultraviolet spectrum is the ultraviolet C, which is around 200 nanometers, which is the border of most conventional spectrophotometers. Most spectrophotometers work around this region, so we're working at the very, very low with far ultraviolet, which is where I observe the effects. And you need a research grade spectrophotometer to do this work because most and many spectrophotometers are not very accurate in this range. This is just the border of where they start, and to get good reproducible readings you need to have a pretty sophisticated spectrophotometer. Okay, so here's a typical tracing from 3 controls. They're virtually superimposable. They can see that they vary a little bit here, but the controls are pretty constant. Here's an example of a classical electromagnetic field. I use 43 kilohertz because it's one of the resonance frequencies of water. And you can see that the effect that we observe is primarily with the amplitude only, and there's a relatively small effect, but work. In contrast, here's a homeopathic remedy where you can see the pattern information is very different. You get a much larger effect at the peak, but now you're starting to get effects, shoulder effects, and then you even start seeing larger effects around 230 nanometers. And this example, the remedy shows a decrease in ultraviolet absorption. Using a laser, you get a similar pattern, but there's no effect at the peak, but there are shoulder effects which show an increase. So depending on the kind of energy we're working with, we get a different pattern. Here's an example of a non-classical electromagnetic field, and since in these days I wasn't sure what frequency to use, so I scanned a wide range of frequencies, and you can see that there's a large effect at the peak, a shoulder effect around 210 nanometers, and then another effect around 230, showing an increase in the absorption. So to summarize all this work, there are 3 patterns that are generated depending on the type of energy source. You either get a primary effect with the peak, you get a shoulder effect, or you get an effect at around 230 nanometers. Now, then I get, this is the conventional or normal observation, but then some energy sources generate very unusual patterns. Not this, not a good example. But here's a vortex, just simply vortexing the water before and after, which we all know affects the water, but you can measure it by this method. So an example of an unconventional pattern would be something like this, which is actually a healer. And I'm not really talking about healers, but you can see the difference in the kind of pattern observed in this experiment. Now, here's an interesting experiment where I use the Mobius strip in the Tesla watch developed by Andrea Puharich, which in itself caused a very large effect and sort of a crisscross over here. But then the watch was then treated with the healer, and we have a combination of, this one over here, is a combination of non-classical electromagnetic energy and bioenergy from a healer. So this is an unusual response. Just wanted to show you, again, that different kinds of energies generate different patterns. Okay, now using, again, the same method for measurement, I wanted to know how long these effects last. This effect was generated using a 43 kilohertz non-classical electromagnetic field, and you can see that the effect immediately after treatment is relatively large, and the 2 samples were maintained over time. One was put in a mu metal shield, and one was just left in the air. So obviously the mu metal shielding, eventually they both go back to control after about 25 days, but the mu metal shielding obviously keeps the energy and the information a lot longer. Okay, so now I want to talk about some experiments with a BIFILLAR solenoid coil where the current strands flow in opposite direction, canceling the current, generating a non-classical field. Again, we're using 43, 44 kilohertz, and in this experiment I actually wanted to compare a solenoid that was in the cancellation mode over here and one that was a classical solenoid where the current is parallel. And the target was water. And the treatment time is 90 minutes. And in order to maintain a similar output from both types of coils, I had to adjust the input voltage so that the output was the same because the resistance of these coils was different. Now, this is an experiment I did in collaboration with Bill Tiller, who did the mathematical calculations for me, and he calculated the intensity of the magnetic field. And in the cancellation mode, you expect the magnetic field to be 0. Well, it was pretty small, 10 to the minus 12 Tesla. He also calculated the strength of the A field. That's the magnetic vector potential. That's the field that generated from a toroid. In this situation, the A field intensity was about 2 orders of magnitude less than the threshold required to imprint water according to Cyril Smith's data. So in essence, there is no A field present. There is no B field present. Yet, there is still an effect on the water as measured with ultraviolet spectroscopy. This slide shows the absorption at 224 nanometers, and the electromagnetic coil, where the non-cancellation in this particular experiment had no effect, and the cancellation solenoid produced a relatively large effect in the absence of an A field and a B field. So this is kind of important observation, extending Cyril Smith's work, indicating that the electrostatic scalar potential, or the phi field, as it's called in physics, also imprints water. Okay, so that's an important observation. Now we're moving on to a different measurement parameter, measuring the optical properties, electrical properties. Standard conductivity measures microsiemens per centimeter at a fixed frequency. I know these little meters that you can buy, they have a fixed frequency. But the techniques I have been using involve what's simply called current voltage relationships. You apply a voltage spike, you excite the water, and you measure the induced current. Now, this concept is used in nonlinear dielectric spectroscopy and impedance spectroscopy and electrochemical impedance. And what I did was, I've done some experiments with this method because it allows you to adjust the frequency. So in experiments we'll talk about later with electrochemical impedance, I adjust the measurement frequency to be the same as the excitation frequency. Then you get interesting and meaningful results with conductivity, in essence, is what this measures. Impedance, resistance, conduction, they're all related. In addition, I've combined these two techniques and developed my own technique, which I call resonance frequency spectroscopy. And we'll get to that in a minute. But before we do that, I basically describing the parameters. Okay, so in these experiments, I'm using signal generator, 43 kilohertz, and signal generators have a lot of different parameters. These are the parameters. Sine wave works best, square wave doesn't work as well, and these parameters using various coils. So here's an example of measurements. Conductivity using the electrochemical impedance, stimulating with 43 kilohertz, and then measuring at 43 kilohertz. And you can see over time, the maximum effect is at 90 minutes, and then it is less if you treat the water for less. So In these experiments, 90 minutes is the optimal treatment time. And overall statistics, if you do it lots of times, it's statistically significant. 24 hours later, the effect drops by 50%, and it's still there, but it's not significant at that point. Okay, so now let's talk about the method that I developed, resonance frequency spectroscopy, which involves electrochemistry of water. Electrochemistry as applied to nonlinear systems like water is well studied by the scientific community, and there are a lot of mathematical theories that to describe the anomalous behavior which occurs in the oscillatory regions. I mean, you get standard linear effects, but then you get these oscillations that occur. And these oscillations are associated with non-equilibrium fluctuations, and this is kind of the scientific explanation of the phenomena. And here's an example of the oscillations that occur from another publication. The kind of oscillations that I get are somewhat different, not as regular, but the problem that the observation is that the current response that I absorb varies enormously in the shape of the spike, the magnitude, the polarity, and the time. So, in other words, at a given moment in time, when you take a measurement, you see a spike, and then you take the measurement a minute later, and the frequency moves. And this phenomenon is called frequency hopping. Frequency hopping is in the scientific literature. Here's an example from IEEE transactions. This particular study talks about frequency hopping patterns and the fact that it occurs across all frequencies, which is interesting because according to Cyril Smith, any frequency can be used to imprint water across the entire spectrum. But the point being that frequency hopping means it's very kind of difficult to get quantitative data. So in order to get quantitative data, the way I analyze the tracings that I get is by measuring the probability of a spike occurring at a particular frequency. So you have to do the experiment 20 times, and then you measure the percent occurrence. How often during those 20 repeat experiments do you get a peak at a specific frequency? So there is the kind of peaks that I get. This is just the raw current at a particular frequency. You see a nice strong positive peak. Here's smaller positive peaks, but the point here is that you get positive and negative peaks. Interesting, but you have to analyze them separately, and then you do the probability measurements, and you get this kind of analysis for the controls, which are about plus and minus 12%. And then if you do an experiment with two different kinds of sources of energy, one being a paramagnetic powder, which I have access to by a company called Altera. And in addition, I placed the cell phone on top of water, assuming that cell phone radiation is harmful. Okay, well you can see that in this is a function of frequency. At the lower frequencies, sometimes you get an increase and sometimes you get a decrease. Where's a nice decrease? Sometimes you get both. But if you look in this particular region, the beneficial mineral only increases conductivity, and the cell phone radiation only inhibits conductivity. So this is apparently the region where all the interesting reproducible phenomena are occurring. And here's an example of water which was treated with a 43 kilohertz non-classical electromagnetic field. And my first question was, can I detect a difference, and then how long does it last? So the effect is still lasting several months after the treatment. So here we have an example of the kind of data from this method, and I've done a lot of other experiments using this method. Here's an example with a technology from a company called, here's another way to generate non-classical energy fields. Instead of using these special coil windings, you can put the coils in different geometries, and you generate what according to Tom Bearden is a superposition of vectors generated from the different individual coils, depending on the geometric arrangement of the coils. Okay, so this concept was used by a company called Fractum, which I'm working with at the present, and some of my colleagues from the company are here, and they have successfully treated water and using it to stimulate the growth of plants, and have some interesting experimental data, field studies with stimulating crop production. So I was interested to what effects that type of non-classical field would have on water, and you can see that depending on the frequency, again, you get very large effects. The largest effect was observed here. This is the treated, and this is the control. So that's the electrical method. Now I want to talk a little bit about my experiments with the GDV, since Paradkov is here at this conference, and I've never really presented this data before. I did in Russia many years ago, but not recently. So instead of putting a finger on the electrodes, we put the water on the electrodes, and this is the kind of experiments. The GDV, where's my data? Oh, I'm missing my data. No, this is the data. Oh, yes, this is the data. That's the measurement. What's the source of the energy? In this case, it was gemstones. Powdered gemstones placed in water. You shape the water, then you filter out the gemstones. The gemstones release an energy, the water picks up the energy, then you measure the energy using the GDV camera. And so in here, for example, is the patterns obtained from 3 different gemstones. This is pH, this is electrical conductivity, standard electrical conductivity with a fixed frequency. And these two correlate more or less, but here's the GDV area, which turned out to be the best parameter. And you can see these kind of measurements, although they do pick up the information from the gemstones, do not correlate with these more conventional methods. So, are we measuring some kind of a new energy with the GDV that's different from conventional? Well, yeah, these methods, electrical energy, pH is not an energetic measurement, but this is a standard method, and this is the GDV camera. Interesting observation. Okay, so that's using the GDV to measure. Now, pH, I have done also recently some experiments with pH measurements. The generator for these experiments was a signal generator with the same parameters that I used before, but no longer using the 43 kilohertz. The purpose here was to, well, if you're going to measure pH, the best frequencies to put in the water would be the frequencies of hydrogen. So what are the frequencies of hydrogen? Well, you could actually get that from various published spectroscopic data. And then you have to, which is the frequency is in the terahertz. So I don't have a terahertz generator. So I had to generate subharmonic frequencies corresponding to the original frequency, and then use those to imprint the water. Now the results are complicated, and I don't really have time to go into that. Maybe next year I can talk about that. I'm using a standard pH meter, so if you do it right, you can get reproducible data, but the results, or depending on the frequency and the combination of frequencies, there are 7 main peaks in the spectrographs of hydrogen atom. And those 7 frequencies were used sequentially to imprint water. Again, at 1 hour, 90 minutes per frequency. Now, depending on the sequence of the frequencies, and depending on which frequencies of those 7 you use, you either increase the pH or decrease the pH. And the results are very complex, and I don't really have time, and my 30 minutes is up, so thank you very much. I thought it would be best to leave time for questions.
Thank you very much. Thank you very much. We go for question first.
Can I have the mic, please?
Yes.
I'm fascinated. The molecular resonances of water, as you say, are in the gigahertz and terahertz region. Could you explain exactly what this subharmonic theory is that you're talking about?
Well, okay. So you have a number in hertz, which is like 10 to the 12th. And there are 2 ways that you can generate a subharmonic. You can either divide by 10, or you could divide by 2. And you eventually work your way down into the kilohertz, megahertz, kilohertz, or hertz. It turns out that kilohertz, and with my system, kilohertz frequencies work best. But I did a lot of explorations and thinking about that. I thought that maybe we need to go all the way down to the fundamental frequency. Yesterday we heard about overtone spectroscopy, where the fundamental frequency is not used, but higher harmonics are used. So I'm thinking, oh, well, let's go down to the fundamental frequency, which would be the frequencies close to 1 hertz. Well, when I did that, it didn't work, so then I went to kilohertz, and I started to see effects. But that is the short answer. We can talk about it later.
Okay. Go back.
So I'll just go first.
You got, yeah.
I had a question about, in one of the first slides, you said that the signal was very complex when you chose for the modified coil. So what was the method to choose the signal parameters?
The first slide about what?
In one of the first slides you mentioned about your experiment, whereby the parameters, the input parameters were very complex, you said.
Oh, the signal generator parameters. The trial and error. I mean, when you, as a scientist, if I'm working with something basic like a signal generator and a coil, and I'm not using one of your technologies, I'm going like, oh, I don't know, what are the parameters? You know, I got a lot of signal generator. You have a lot of different parameters. So all trial and error, and you know, how much AC, how much DC. I mean, signal generators are complex, and you can generate a lot of different types of waveforms and signals, and it was all trial and error.
Okay, I have a two-part question. First part is, in the beginning you said that non-classical fields precede biochemistry.
Yes.
If you maybe could elucidate a little bit on that. And part B of my question would be, what is your opinion or point of view on the Cyril Smith's multiple frequency effects?
The first question was about
non-classical fields precede biochemistry.
Right, yes. Okay, so it's well established that classical electromagnetic fields regulate biochemical processes. The Bioelectromagnetics Society is full of examples where an electromagnetic field can influence and modulate a biochemical process. In my model, I believe that non-classical electromagnetic fields precede classical fields because they underlie classical fields. The non-classical, you take a classical field, you cancel it, and what's left underneath is a non-classical field. So it's a more fundamental type of energy. So I believe that the sequence is that, actually I would put quantum fields first, which are more fundamental, which regulate non-classical fields, which regulate classical fields, which regulate biochemistry. That's my model, my working model. Okay, and the second question about...
Was the Cyril-Smith multiple frequency effect?
Yeah, well, I have worked with Cyril Smith, and although he uses subjective methods to analyze the water, I prefer to use quantitative methods, and you saw some of the examples. But I believe in the results that Cyril Smith has collected because when we worked together, I also used his method, which is basically radiesthesia or a pendulum, and I obtained the same results that he did. So that's probably maybe the only person side by side, because he was there. So his energy was there, his consciousness was there, his influence was there. He wasn't holding my hand, but he was next to me. So I'm doing the pendulum, and I got the same results. So I believe his data.
Thank you very much. Thank you very much.